Hi, my name is Henrike Krenz, and I'm a PhD student at the Institute of Medical Informatics of the University of Münster. I'd like to share my new project, the myGeneAtlas, with you, which is a template for a disease-specific gene browser application. You might be thinking right now that there's already a multitude of genomic databases and web applications available on the internet, and you're totally right with that. So these are only a few examples. Some of them are focusing just on human genes and clinical and functional information about them, on specific bioinformatic techniques like expression data, exome or genome sequencing data, or genome-wide association studies. And others might be focusing only on specific diseases, like a specific type of cancer. However, there are always some rare diseases that are not sufficiently represented among these applications. And for many researchers in the fields of biology or medicine, it is really difficult to come up with their own web pages for these specific rare diseases. One of these diseases, which is not sufficiently represented among these web applications, is male infertility. Therefore, we recently developed and implemented the male fertility gene atlas and published it on the internet. It's basically collecting publications on studies for male infertility and germ cells and presents them in one place, such that researchers can easily find them. All of these studies contain data about genomic, transcriptomic, or methylomic studies. Male fertility gene atlas can record all of these different types of data. For this application, we had two main requirements. The first requirement is that relevant publications should be retrievable based on conditions, tissues, cell types, gene names, and some other search terms that should be specified by the user. Most importantly, data tables, images, and supplemental data of publications should be searched for mentioning of the search terms as well. This is an example screenshot for searching the MFGA database for the conditions azoospermia, obstructive azoospermia, and non-obstructive azoospermia. The second requirement is that there should be aggregated information over all relevant publications that have been found, like, for example, genes that are mentioned most frequently in publications about a specific condition or cell type. This here is an example of the genes that are mentioned in publications about azoospermia. As you might have noticed, these requirements are not specific for male infertility. They could as well be used for any other disease. So the concept of the male fertility gene atlas could easily be generalized. We are planning to transform the code of the male fertility gene atlas into a template for a disease-specific gene browser application. With this template, researchers from other fields in biology and medicine can create their own gene browser applications. The architecture of this template will be the same as the architecture of the male fertility gene atlas. However, the data model will be further optimized, such that the search functions will be easier and quicker, and such that there's more room for aggregated information. Right now, MFGA employs some external libraries that are not open source. These have, of course, to be replaced for a template with open source resources. The user interface of the male fertility gene atlas is already based on the Bootstrap library. It is enabling a good representation of web pages across different browsers and devices. However, for myGeneAtlas, we would like to use Bootstrap more consequently. Finally, everyone will be able to use our template because it will be published on GitHub as an open source software. In order to give you an impression of how myGeneAtlas might look like, I will show you two screenshots in the following. This here is the header. The header might of course be adjusted to each of the diseases that might be represented via myGeneAtlas. Additionally, you can see here some aggregated information about the example data that is in the myGeneAtlas database. So, for example, some basic statistics about how many publications and datasets are present, or the top genes mentioned in the database. Also, there is a plot showing which omics types are currently available in the database. Thank you for your attention. If there are any questions on the project, please feel free to contact me via email.